Hi, my name is Jacob Rimlaria, a rising senior at Hawken. I worked in Dr. Gerson's lab with the Case Comprehensive Cancer Center for the topic of TMZ, temozolomide, in cancer therapy. My project is possibilities of combination therapy with TMZ, mutated MGMT, and DNA damage response inhibitors. Temozolomide, or TMZ, is an alkylating agent and methylating agent used in cancer therapy. Different methylating agents have specific affinities and specificities to certain nucleophilic sites. Each base has several nucleophilic sites. TMZ produces N7-MG and 3-MA and O6-MG toxic adducts through adding methyl groups to the different bases guanine and adenine at the N7 position of guanine or different positions, producing these several adducts. N7-MG and N3-MA are more abundant than O6-MG, but are less cytotoxic and are repaired by base excision repair, BER, while the O6-MG adducts are repaired by the O6-methylguanine DNA-alkyltransferase, MGMT repair enzyme. As I just mentioned, the N7-MG and N3-MA adducts created by TMZ are usually repaired by BER, base excision repair. DNA glycosylases initiate the BER pathway by recognizing excising damaged bases and creating a basic site. The glycosylases break the N bonding with the bases through hydrolysis using the common intermediate between BER glycosylases AP sites. Apurinic endonuclease 1, APE1, recognizes the A basic sites and cleaves around the end of the DNA. Then BER splits into two different pathways, either long patch BER to replace a group of nucleotides, or a short patch BER to replace one nucleotide. The N7-MG and N3-MA toxic adducts are repaired by the short patch BER, where DNA polymerase beta, polybeta, along with the XRCC1 ligase 3, Lig3 complex, complete the repair process. Targeting the base excision repair pathway in combination with TMZ is a good plan and pathway for sensitizing cancer cells to TMZ. MGM2 enzyme transcribes the AGT protein, which then repairs the O6-MG toxic adduct created by TMZ. The O6-alkylguanine DNA-alkyltransferase, AGT protein, scans double-stranded DNA for alkylation at the O6 position of guanine and then directly transfers the methyl group from O6-MG to the AGT protein, which inactivates the AGT permanently. MGMT is a good target for inhibition for cancer therapy in combination with TMZ. Some mutations in MGMT have been identified, resulting in AGT repair proteins that are actually resistant to inhibition. The MGMT mutant genes can be used in gene therapy to decrease the toxic side effects and protect the healthy tissues, while still allowing the cytotoxicity in tumor cells. TMZ is a very useful alkylating agent that can be used in combination therapy with inhibitors of the DNA damage response and with mutated MGMT to get increased cytotoxicity while sparing healthy cells with gene therapy. The goal of this project is to look at the different repair pathways involved with TMZ and explore how these pathways can be used to increase the cytotoxicity of TMZ. Another objective is to look at gene therapy approaches using mutated MGMT enzymes to reduce the toxic side effects of alkylating agents. My hypothesis is that when combining temozolomide, TMZ, with inhibitors of the cell's repair response to TMZ, the cell will be sensitized to TMZ as the repair response to TMZ will be halted, decreasing the cell's resistance. Combining TMZ with the mutated MGMT will decrease the toxic side effects of TMZ by endowing the Hematopoietic stem cells, HSCs, with an increased resistance to MGMT inhibitors. To test this hypothesis, I did a literature review and read various reviews and research papers related to the topic, who found their information from experimenting themselves or from others' papers. They did research through exploring cell lines, testing on mice, and clinical trials done with TMZ and other drugs in combination therapy. I researched the topic by reading qualified research papers, and reviews written about the topic that were developed from research done. In various reviews, different basic excision repair inhibitors were tested with TMZ, since TMZ induces N7-MG and N3-MA adducts that are usually repaired by BER. Methoxamine MX, a BER inhibitor, binds to AP sites created by the BER pathway and reduces the APE cleavage of the backbone, therefore increasing the cytotoxicity of TMZ by disrupting the BER pathways. Poly-ADP-ribose polymerase, aka PARP, is a regulator of the BER pathways, as PARPs consist of enzymes that transfer the ADP-ribose to target proteins, activating and playing a really important role in the BER pathway. So a combination of TMZ and PARP inhibitors enhance the cytotoxic effect of TMZ, and this has been shown in glioblastoma preclinical models through the inhibition of the BER pathway that PARP inhibitors do. O6-MG adducts are also induced by TMZ, but they are normally repaired by MGMT. MGMT inhibitors have been developed, including O6-benzylguanine, or BG, but BG has been shown to inhibit MGMT repair of O6-MG adducts and therefore enhance the cytotoxicity of TMZ, but when it was used in trials with TMZ, it was determined that it had a severe myelosuppression toxic side effect. The mutated MGMT that was discussed earlier is when transferred into the hematopoietic stem cells, results in the hematopoietic stem cell resistance to BG and TMZ treatment, which minimizes the toxic side effects of BG and TMZ treatment by leaving their MGMT repair enzyme intact. And since it cannot be inhibited by the BG, and its repair pathway of TMZ is still intact. TMZ had an increased cytotoxicity when combined with different BER inhibitors and MGMT inhibitors, and its toxic side effects were minimized through gene therapy, implanting mutated MGMT into hematopoietic stem cells, increasing the cellular resistance of those healthy cells to MGMT inhibitors. This review dealt with the uses of DNA damage response inhibitors and gene therapy, along with TMZ in cancer therapy. It focused on a few inhibitors, MX and PRP inhibitor, which are both inhibitors of the BER pathway, and BG and MGMT inhibitor, which were all used to give TMZ an increased cytotoxicity. TMZ was also used with BG and the mutated MGMT gene, which gave TMZ an increased cytotoxicity to the normal cancer cells, but it still spared the healthy cells, since the HSCs, hematopoietic stem cells, were endowed with the mutated MGMT gene, giving it an increased resistance to BG inhibition of the MGMT gene. The success of these drugs and models have very good implications for the future of cancer therapy. It brings up the possibilities of gene therapy and increasing the resistance to certain drugs to spare certain cells by implanting specific mutated genes within different stem cells. And it also speaks of many drugs that would be useful with different genotoxic agents that are usually repaired by BR, basic excision repair, but with the discussed BR inhibitors, it would have an increased cytotoxicity. The success of the research papers and reviews should transition over to testing these inhibitors and mutated MGMT genes in clinical trials to see if these results work in humans as much of it has worked in vitro and with mice trials. I would like to thank Dr. Quinn for helping me throughout the summer program. I would also like to thank Dr. Gerson and the whole entire Gerson lab for sponsoring me this summer and meeting with me to review different readings and having good discussions where I'm able to learn much new information. That's really interesting to me.